he's working as a postgraduate teacher with the Savitri Bai Phule Pune University in India, and he received a funding of 150,000 rupees for a project development and evaluation of floating drug delivery systems by novel approaches. And he's from Pune. He's also had published a book called as Biodiesel from Waste Cooking Oil, a Potential Alternative Fuel in the Lambert Academic Publication, Germany. And he's working as an international reviewer for the American Journal of Advanced Drug Delivery. He has 15 publications to his credit. And today he's going to speak about adverse drug reactions. It's a threat more to children and women than adult male. Sagar.
Yeah, good afternoon, everyone. My topic is on ADR is a threat more to children and women than adult men. Well, that ADR is majorly a health problem to the society, and according to the WHO definition, it's a response to a drug which is noxious and unintended and which occurs at doses normally used in man or prophylaxis, diagnosis or therapy of disease or the modification of physiological changes. Then, amongst the known risk factors are increasing age, polypharmacy, liver and renal diseases, as well as being a female. Yeah, female patients have 1.5 to 1.7 fold greater risk for developing of an ADR, including adverse skin reactions compared with male patients. The reason for this increased risk are not entirely clear, but include gender-related differences in pharmacokinetics, immunological and hormonal factors, as well as differences in the use of medications as compared with the men. Women generally have lower lean body mass, a reduced hepatic clearance, have differences in activity of cytochrome P450 enzymes, that which 14 increases in CYP3A4, where decreases in CYP2D6, CYP2C19 and CYP1482, and metabolized drug at other different rates compared with men. Other important factors include conjugation, absorption, protein binding and relay elimination, which may all have some gender-based differences. These are some pharmacodynamic differences between men and women, seen particularly with cardiac and psychotropic medications. It is possible that the gender differences in T cell activation and proliferation account for this, as well as it increased prevalence of skin disease such as systemic lupus erythematosus and photosensitivity. Whatever the mechanisms, it is important to be aware that gender is a significant factor in ADRs. In analysis of 48 community-based cohort studies from the UK, the overall incidence suspected of ADR in males was 12.9 per 10,000 patients months of exposure, and in females was 20.6 per 10,000 patients in months of exposure. The overall age standard odds ratio of an ADR in females compared with males was 1.6, means 95% confidence interval C1, 1.5 to 1.7. This gender difference was significant in all age groups above 19 years of age and was relatively constant across all age of groups. In the report of the Spanish system of pharmacovigilance, 60% of 1609 adverse reactions to non-steroidal anti-inflammatory drugs were in women, that's odds 1.67. And in further Canadian study, it's 74.1% of ADRs were in women. Drug classes most frequently reported to elicit an adverse event were general anti-infectiveness, nervous system agents, musculoskeletal agents, skin related reactions accounted for the all reported ADRs. More than one agent was reported to be responsible for the ADRs in 50% of the female patients, compared with only 33.1% of all male patients. In a prospective studies of 1920 hospitalized patients in Chile, the frequency of ADR was significantly higher females than in males. And in larger Italian study of ADRs in a population of over 20 million people, cutaneous ADR made up of 45% of all reports. These are several gender-based differences in both drug pharmacokinetic and pharmacodynamics. Differences in lean body masses are important for some drugs. Example, diazepam, where women have higher volume of distribution compared with the men, and alcohol, where the reverse occurs. A number of studies suggest that cytochrome P450 3A4 activity is higher by up to 40% in women, important enzymes compared with men, and CYP34A4 is probably the most important enzyme associated with drug metabolism accounting for perhaps 50% of metabolism of therapeutic drugs. Examples of increased CYP34A activity include erythromycin, which is metabolized 25% more rapidly in women. Then the gender difference in steroid hormones influencing CYP3A4 activity, that is estrogen and progesterone mainly, which may act competitive inhibitors. Most other enzyme systems involved in drug metabolism, example CYP2C6, CYP2C19 and CYP182, appear to be more active in men than women, although results from studies are not consistent. The effects of gender on pharmacodynamics are less clear. Women are at increased risk of QT prolongation with certain antiarrhythmic drugs compared with men, even at equivalent serum concentrations. There is no doubt that number of psychotropic drugs including chloromazepam, fluoxetine and various antipsychotics appear more effective in women than men for the same dosage. Idiosyncratic drug reactions, particularly cutaneous reactions, appear to have an immunological etiology. The onset of drug hypersensitivity involves drug bioactivation, covalent binding to proteins, followed by uptake, antigen processing and T cell proliferation, whilst drug metabolism plays a vital role within the balance between metabolic and bioactivation and detoxification being an important component. T cell activation and proliferation may be as important. However, well organized that women have increased incidence of a number of skin diseases thought to have immunological diseases basis. These include systemic lupus, erythrocytosis, systematic sclerosis, photosensitivity and lichen plants. Women are significant different range of drugs than men, particularly drugs associated with oral contraception, the menopause and pregnancy. However, the observation that women consume significantly more over-the-counter medications, herbal remedies and vitamins compared with men suggests that access to healthcare is unlikely to explain the differences. The ADR in children is the most important for this. Adverse drug reactions in children are more important public health problem. We have undertaken a systematic review of observational studies in children. Criteria for considering studies for this review included studies like observational studies that estimate the incidence of ADRs, including retrospective and prospective cohort studies of children. Excluded studies which focus on ADRs in relation to specific drugs, example antibiotics and or carbamazepine. Clinical condition, example epilepsy and asthma, or specifically clinical presentations of ADRs analgesics. Case control studies, those carried out exclusively on a neonatal intensive care unit. Studies reporting medications errors, therapeutic failures, non-compliance, accidental and intentional poisoning and drug basis. Participants, studies included three defined population. 1. Children admitted to hospital, children in hospital and children within the community. Incident rates of ADR causing hospital admission ranged to from 0.4 to 10.3 of all children and 0.6% to 16.8 of all children exposed to a drug during hospital stay. Anti-infectives and anti-epileptics were the most frequently reported therapeutic class associated with ADRs in children admitted to hospital. 17 studies were carried out of 12 studies respectively, and children in hospital 20 studies carried out of 14 studies respectively. While anti-infectives and non-steroidal anti-inflammatory drugs NSAIDs were frequently reported as an associated with ADRs in outpatient children. 13 studies carried out, 6 studies respectively. That 14 studies reported rates ranging from 7 to 98% of ADRs being either definitely possible available. We have included 51 studies where ADRs have been investigated in the hospital setting. The period under study varied widely and ranged from 1 day to 10 years. The majority of studies were described as being performed in a general pediatric unit or ward. 2, which is included in intensive card also. 6 studies were performed solely in the intensive care setting, one of which included general medicines. 3 studies included children on isolation ward. 1 study was performed using an integrated primary care information database and 1 in an isolation ward. The remaining 13 studies were covered a combination of clinical settings. In an agreement with previous studies, including those specific to adults, this review found the ADR incidence rates were generally higher in hospitalized children than ADR rates causing hospital admission or in an outpatient settings. In September 2013, the European Medicine Agency (EMA) Pharmacovigilance Risk Assessment Committee, that is PRAC, initiated a safety review on exophthalmic dermatitis associated with ustekinumab. This signal was identified through routine pharmacovigilance surveillance in the UK, where 12 cases of exophthalmic dermatitis and 15 cases of erythrodermic associated with ustekinumab treatment were received. In November 2014, Health Canada issued a safety review summary highlighting the possibly link between exophthalmic dermatitis and the same. In the safety review, Health Canada reported that it had received 5 reports of skin exfoliation and non-serious report of exophthalmic dermatitis associated with use of ustekinumab. In 2014, the Vigilance and Compliance branch of HSA captured a total of 2170. Thanks. Local adverse event AE reports suspected to be related to health products. The breakdown of the number of valid reports captured in the National AE database from 2006 to 2014 based on the data received is illustrated in figure 1. That is, number of AE reports captured in database from year 2006 and the number of AE reports to captured. There were more AE reports received for females 54% than males 43%, and 3% reports did not indicate the gender of the patients. In 2014, HSA received a total of 249 AE reports suspected to be associated with vaccines, of which 209 reports 48% involved children aged 12 years and below, which corresponds to the age group of vaccines under the National Childhood Immunization Schedule. Of these, 84 of the reports and is equal to 176 were captured by KK Women's and Children's Hospital KKH Active Surveillance Sentinel site. A total of 102 studies were included in the review. 80 studies described in the clinical event as an ADR. Some studies included multiple settings. 42 studies investigated ADRs as the cause of admissions to hospital. 51 studies investigated ADRs in the hospital settings and 36 studies investigated ADRs in the community settings. Studies included in over review were conducted in 31 different countries, mostly Europe and America. ADR detection methods were employed in 58 out of 102 studies. These consist of a combination of case record review, drug chart review, laboratory data, computerized ADR reporting system, attendance at ward rounds, and interviewing patients, parents, clinicians. In 31 studies, case record review alone was undertaken. The remaining 11 studies used parental interviews, questionnaires, 5 studies, clinical assessments, clinical questionnaires, ward round, and a nationwide computer database The remaining study report did not refer to the methods used. Conclusions. Women certainly appear to have a significantly higher risk of developing adverse drug reactions than men. This appears to be because of a number of gender-based psychological differences as well as differences in the use of medications between men and women. This study has shown ADRs to be a significant problem in children and has highlighted therapeutic classes of drugs most commonly associated with them. Therefore, availability of ADRs is needed. Further, women on how much ADRs may be prevented should be carried out in recent years. These are the references from Is there any questions?
I have a question on the fact that you actually name a couple of products that have a higher risk of causing adverse events in females. I was wondering, would you know how often a package insert or a summary of product characteristic would have specific dosing requirements for women as compared to men? How often is that taken into account?
No, it's all the given data by some of the reviews, na? So that's why I included in that.
In the summary of product characteristics, there would be dosing recommendations for women that would differ than for men?
No, yet till they have not mentioned, any company yet till not mentioned, but it's the conclusion I have given.
I have a question, two questions. The first one, you've focused a lot on the physiological differences between men and women in terms of metabolism and other drugs that are used. I was wondering, did you look at any possible social factors as to why that would happen? So would women be more inclined to report adverse drug reactions than men? Just one example. And also, although there is a relative risk of an increased chance of an ADR in women than men, was any of the data sort of stratified by severity or seriousness of those events?
Yeah, there is a basic difference in the structure of human and men's body. So there are some enzymes, those are sprinkled in women's and not useful in men's. So it's the basic difference.
You mentioned something about the immunological differences. Can you please tell me something about that? You're talking about cutaneous reactions. So how does the immunology of the lady or the immunological response differ from that of a male? Yeah, did you find any changes in the T cell responses?
No, sir. Sorry.
No.
It's quite interesting actually because you also mentioned systemic lupus, SLE,
Yeah.
as having a higher incidence. Adverse drug reactions of SLE has a higher incidence in women than as of men, but in the general population, women have a higher. SLE is generally higher in the female population than in the male population. So I was wondering if there was any. What sort of biases did you have? Did you account for in this at all, if you did?
Okay. That's what he's saying, that's T cell activation profilation account, this one, na?
Yeah, it's all going back to the T cell because dendrites and things there causing a cellular immune response. Is there some difference between the T cell response in a male and a female, or you haven't studied?
No, I haven't studied, sir. Yeah.
Is it an actual research that you conducted, or is it a review of the research?
Yeah, it's a review of all the research I have given. Yeah.
Thank you very much.
Thanks.